Hi guys and welcome to Learn Extra Live. It is a Wednesday, that must mean it is life science and grade 10s. I hope that you are tuning in. As you can see, I've got Ntokozo, or known as Knox, one of our top 10s here in studio. How are you, dude?
I'm finding yourself, indeed.
So good. So nice to see you in studio.
I'm really excited to be here, and I'm really hoping that the guys at home are ready for the wonderful and awesome lesson that we have in store for them.
I know. I can't wait. So I just wanted to give a big congratulations to you to making it to the top 10.
Thank you very much.
Yeah, and you guys all voted this guy in. So big thank you to you as well.
Thank you.
So now before we start, Knox, tell us a little bit about yourself.
Okay, so I'm 18. I'm a law and international relations student at the University of Witwatersrand. That is in Braamfontein. And yeah, I'm a cool guy, very easy to talk to and very easy to approach.
And where do you come from?
Originally, I'm from Soweto in Diepkloof.
That's amazing, hey? So you're gonna defend me if I ever get into any sticky situations.
I'm pretty sure I will. I'll try.
Yes, guys, everyone needs a lawyer friend in their lives. I'm telling you. Hey? Okay, so Knox, tell them where they can find us.
Okay, so you guys at home can go to www.facebook.com/learn extra, and that's where you can get in contact with us.
Exactly. So post all of your questions on the page. For grade 10, Knox is going to be here on the page answering all of your questions. And guys, if you're looking at this little pack here and you're wondering what are all of these amazing goodies, this Cheetos pack, we are doing a promotion or more, like a little incentive or competition. It's called Random Acts of Kindness. And what you can do is that you go and you post your random acts of kindness onto our Pay It Forward events page, and you could win a hamper from Simba with all these Cheetos goodies, this Cheetos little notebook, these awesome CDs. I think there's 1, 2, 3, 4, 5, and it's Donald's, Train of Love, Unati. We've got Kaya for you. You kind of look like Kaya, actually.
No, really? Do I?
Malik. And the only love songs album you will ever need. That's what it says right there. And guys, random acts of kindness don't take money. It's about the small things. It's about helping your brother or sister with your homework, carrying the shopping for your mom. It doesn't take money. It's more about giving and helping. That's exactly what it's about. So now we've actually got Eugen with us today, and he has got such an exciting show planned for you, Life Science. I think let's actually go straight to Eugen. I can't wait. I see Cells Alive. I love Cells Alive. How are you, Eugen?
Good, and you, Indy? How are you doing?
Good.
Good, guys. Guys, welcome to Life Sciences Grade 10 lesson. So today we're going to look at cells. We're going to do a bit of the microscope, but we're going to look at the details of an animal cell during this lesson. We're going to have a bit of looking at how to use a microscope and the parts of the microscope. But before we get into that, let's look at what's in store for today's lesson, the key points that we're going to do, and then we get on to the details of grade 10 life sciences and cell biology. So very quickly, we're going to be looking at cells today, and I've got a nice picture of a cell here. This is an interactive site. I'm going to show you the link to that a little later, but we're going to refer to this constantly through the program. And let's get to the lesson presentation. So we're looking at animal cells today. Okay, so let's look at. So in this lesson, we're going to be studying the cell theory. We're going to look at a list of terminology and relevant terms that are important to cell biology. We're also going to be dealing with parts of the microscope. We're going to look at a definition of a cell and what a cell is. We're going to look at some examples of cells, types of cells. We're going to look at some plants and animal cells very superficially, because we're going to spend lots of time today looking at the animal cells, the organelles, and obviously the parts and the structure of these organelles. So let's move on to the let's move on to the cell theory. So guys, you've got to think back in terms of when the microscope was discovered. And remember that we're not going much into the details of the microscope today, but we're going to look at what the cell theory is. And basically the cell theory it talks about all living things being made up of cells. So when we talk of living things, all living things, guys, are made up of cells. And these cells or these living things could be either unicellular or multicellular, which simply means uni meaning made up of a single cell, or multicellular meaning made up of multiple cells. So we're dealing with organisms that are microscopic or that can be seen to a limited extent with an aided vision. So we're using a lens, a hand lens, or a dissecting microscope to see the details. So that is basically what the cell theory deals with. Cells are the smallest working units of living things, and they show the characteristics and properties of life. Now, what that simply means that in order for us to classify something as living, it needs to show the characteristics of life, and which are simply being able to reproduce, being able to respire, be able to respond to a stimulus or the environment, being able to produce waste, being able to move. That's important because these are characteristics of all living things, and hence we call a cell the smallest functional unit in an organism. All cells come from a pre-existing cell through cell division. So in grade 12, when we look at evolution, we look at how organisms have evolved, and we talk of a primordial cell or an ancestral cell. And we say in life sciences that all life forms have evolved from a primordial universal ancestor, which was unicellular. And that gave rise to complex organisms that became eventually multicellular as we have today. So we will look at how living things compare in size next. So when we talk of living things comparing in size, we refer to organisms being microscopic, and we refer to some being submicroscopic, meaning those that cannot be seen even with the conventional light microscopes that you guys have in your labs. You would need an electron microscope, and for that, most of our schools do not have these, and you probably get into, when you're doing research, you'll probably have access to an electron microscope. And then we have organisms that can be seen with the naked eye. And if we look at this diagram here, basically it shows you a scale of organisms from submicroscopic. Here we've got organelles, and here we've got a cell, and that is about 10 micrometers in diameter. And here we have humans, which could be between 1 meter to 1.5 meters average height. And then we have trees that could increase in length to about a kilometer in height. So when we look at organisms, when you look at living things, the ratio and size of these in relation to each other vary, and that's important for us to refer to. I'm going to click on a link very quickly for you guys to show you how big things are in relation to each other. When we look at this interactive, and you guys can find the link at the end of the notes to this. Here
I'll also put it on Facebook. I love Cells Alive.
So super, you guys can try this out. It's quite interactive, and it's about trying it out. And here we have, on a needle, we have a strand of hair. I'm going to go back, guys, to show you. Okay, so that's, okay, it's obviously not moving back there. Okay, so there's, okay, so as we move in gradually into this, we can see that's a pinhead, and as we move, there we have a hair follicle sitting on a pinhead. If we go greater into the details, we see on that we've got some microorganisms that are there, but we're going to find and look at. So that is at a magnification of almost 100 times greater in size. If we go further down and start increasing the magnification, here we see some cells. And if we talk of dust mites, here's a dust mite, and these are the dust mites that you'd find in your carpets, and sometimes they are quite big in relation to submicroscopic organisms, and that is what you'll be able to see with a hand lens or dissecting microscope. You do not necessarily need a light microscope to see a dust mite because it's much larger. If we go further, guys, further down, we're going down. This is about 1,000 times now, so we magnify these objects 1,000 times. Here we see a red blood cell, guys. Here we see a red blood cell, and here we're going to look at, so we looked at the dust mite. Here we're seeing pollen, and this is the pollen that we have on plants, and it's from a ragweed plant, and here you can see in relation to a red blood cell, its size. We can go further down into a red blood cell, and that's a red blood cell right here, showing us its size in relation to a ragweed pollen. If we go further down into a lymphocyte, now lymphocytes are cells that we will do later on when we look at composition of blood cells. You would see that lymphocytes are much larger than red blood cells. But again, if we look at something like an E. coli, guys, and these are E. coli here. Here, if we see E. coli, guys, these are the E. coli. Now, these are bacteria that are found around us, and they play a very important role. They're found in the digestive tract, and here you can see in relation to a red blood cell, they're extremely small. And this is a scale showing us that this represents 2 micrometers in length. So if we look at a red blood cell, it's much larger than 2 micrometers in length, but if we look at a bacterial cell, in relation to that, it's almost 2 microns in length. If we go further down to looking at viruses, which are much smaller than red blood cells, here we're seeing the Ebola virus, which is this here. If you look at that, and that is 200 nanometers in length. So we can increase that. We can go further down and look at a rhinovirus, which is one of the smallest viruses that you possibly would see. And there's the magnification here. So these are extremely small. So you can play around with this, guys. It's a link to this is on is attached to the notes. It's on Facebook as well. You can it's quite interesting to play around with these different learning tools. And remember, the more you interact with these, especially with a topic like cells, when you actually cannot see cells in life, you've got to use technology, you've got to use media to try and enhance your learning. And that's essentially what we try to encourage you. We try and give you the best opportunities, the best links to link onto and to enhance learning. Now, and I think this is important, guys, and I've always said this, create a vocabulary list of terms that you constantly refer to. So now I do this at the beginning of any chapter that I teach, and you should do this as well when you're preparing your notes. Create a list of all the terms that are important to you. And these are the terms that we're going to refer to. And during your studying, you can easily refer to them, write on what they mean, so that you can refer to them when you're in doubt or you're not too sure. I'm going to throw Knox in the deep end, and I'm going to ask him what's happening on the page, and if he can give us some feedback on the activity on the page. Knox.
Knox.
Yes, Eugene, thank you very much. We have Karabo Duffy Man, Hollywood Boy Maluka. That's a very long name, by the way.
Karabo, very long.
Very long. It's Karabo Duffy Man.
Well, he hasn't got a question,
Yeah.
and there aren't any questions on the page. So what I'm telling the mindsets to do is, guys, please post your questions on the page. So next time when Knox is dropped in the hot water, he actually has a question to ask. But I said to Knox that he should read this one out anyway.
Sure. Karabo says that mindsetters, please help me grow my page. My username for the page is www.facebook.com/distinction room. Just in case, please.
Yeah, so Karabo, if you missed that, guys, yesterday he was the winner of the Pay It Forward campaign, and what he did was he started this Facebook page to help other mindsetters with maths and physical science, and it's www.facebook.com/distinction room. We will reshare, and Karabo, big up to you for taking that initiative,
Sure,
right?
I think it's a great one, actually.
Yeah, cool. Thanks,
Awesome.
Eugen.
Awesome stuff. So we're going to continue, guys. So we're going to look at the microscope. Here I've got the parts of the microscope, the various parts labeled on this diagram. And what's good about having a microscope is that we can obviously use it in the lab, but today I have two microscopes that I want to show you, or two different apparatus that actually enhance the ability to look at substances or objects that are submicroscopic or extremely small that cannot be seen with the naked eye. So I'm going to very quickly look at the diagram that we have here that's labeled to show you the parts, and then we get into looking at the actual microscope that we have. So we have obviously a microscope. This is a light microscope, guys. You have an ocular piece, and that's the eyepiece. Here you have the arm that holds the parts together. We have the revolving condenser and the nose piece, which has the different objectives on them. We have the coarse adjustment, the fine adjustment. But I know that these notes are there for you, and that's the reason why I've gone through them. But I want to get over to and showing you how to use a microscope. Not necessarily how to prepare a slide today. We'll do that next week. But just to look at the parts of a microscope so that you understand the parts. And if you asked a question to label the parts and their functions, you understand exactly what these parts are. So let's go and have a look at these. Look at this. So we have two microscopes here, guys. We've got a light microscope. And I'm sure you guys know why it's called a light microscope, because obviously it requires a source of light to be able to work with. And also we have here, and this looks a bit weird, but it's a binocular, it's a dissecting microscope. And when do you use a dissecting microscope? You'll find that often you probably have a seed that you want to look at. You probably would have something that you cannot prepare a slide with. That is a little larger than something that is microscopic. So if we want to look at a hair follicle, we could look at a hair follicle here. If we want to look at a dust mite, we could see a dust mite with this. And this simply means placing a specimen onto so here I'm going to place a coin and onto the stage, and then we adjust we've got a coarse adjustment on this here, and we have a fine adjustment on the ocular pieces, which help us to look at substances or the external structure of substances. So we could look at a leaf, we could look at the stomata of the leaf, and this is quite a very good tool to look at when you're studying mosses and ferns. So you don't really need to prepare a slide. You put on the specimen and you're looking at the external view of the specimen. The light microscope, however, you'll need to prepare a slide. And today I'm not going to spend time preparing slides. I'm going to show you how to prepare a slide next week when we look at plant cells. But I thought that it's important for us to understand how to use a microscope. And very important, guys, when you have a microscope, you've got to place the microscope on a stable, flat surface. And obviously I've got this placed at an angle so that you can see this, but it needs to be placed in such a manner that it faces you and you're behind the arm and the coarse adjustment is closer to you. I'm going to turn this at a slight angle so that you can see when I refer to the parts, the different parts. So we call this a light microscope, obviously because there's a source of light that comes in from a lamp in here that we switch on, and that light strikes through the diaphragm. Okay, so firstly, when we look at a microscope, we look through the microscope through an eyepiece, and that's called an ocular piece. And this obviously has a lens which helps the initial magnification. So this has got a calibration of 10 times. So this will magnify whatever we're seeing at least 10 times. Then we have, this is called the nose piece, and on this nose piece we have different objectives that revolve. And the idea of having different objectives is to be able to look at your specimen under different magnifications. And always when you're preparing a slide, you got to set your microscope on a flat surface, you put your stage down, and then you place on your slide, and then you adjust your objectives to the lowest magnification possible, and then you place on your slide, and then you start observing. So we have your revolving nose piece which have different objectives, and these basically magnify the specimens to a different magnification. If we wanted to see the nucleus of a cell, we would need to use the highest magnification. If we wanted to see the cell wall, guys, you'd probably be able to see the cell wall of plant cells using the lowest magnification. So you're going to place, we're going to do the details. You place your slide on there, and you need to adjust the specimen. You have two types of adjustment. You have a coarse adjustment, guys, and a fine adjustment. And it's very important for you to understand this. When you place your slide on the stage, you need some microscopes have stage clamps that clamp the slide onto the stage. Here we've got also a similar clamp, but this clamp allows for us to move the slide so that we do not have to physically touch the slide once it's on the stage under the objectives. We also see here at the bottom, we see the diaphragm. And guys, the diaphragm is basically there to control the amount of light. And the fundamental concept behind using a microscope is using light that passes through the specimen, and that light falls on your retina, and that is the image of what you're seeing. You'll find that sometimes we do not control the light correctly, and the images that are created are not as clear as we need to. So very important is that you should be able to control the amount of light that falls on the specimen, and that is very important. So you place your slide on there, and you adjust the light, you adjust your coarse adjustment, and then we move on to the fine adjustment. The fine adjustment is used for clearing the accuracy of your image, and often you find it a bit hazy, and you can change that by playing with the fine adjustment. After that you will then change onto a higher magnification, and you probably get onto the highest objective in terms of the strongest power of the objective lens. And when you're on this objective, guys, you got to remember that at this point of time, the space between your slide and your lens is very little. So we're going to go for a break, and when we get back, we're going to look at the parts of the slide.
Okay, guys, it is time for a break, and sadly, in Tokozo, it is time for you to go off the show. But guys, it's okay, because he's in the background. He's going to be answering all of your questions on Facebook, so find him there, and we'll see you guys after the break. Hi guys, and welcome back to Learn Extra Live Grade 10s. This is Life Science, just in case you have missed out. And if you have missed out, that means that you missed the top 10 Ntokozo here on the show. He is awesome, and he is still behind the page writing back to all of your questions. Guys, this is a very, very important link that you need to know throughout the year. It's www.facebook.com/learnextra, that one too. But this is the link to our website. It's www.learnextra.co.za/live. And what that's going to get you, it's going to get you all of our schedules, it's going to get you the show notes, and it's going to get you the previous week's videos. So if you haven't been there yet, go there now and download this lesson's notes. It's pretty awesome. You can work along with Eugen, and in the next 3 days, we will be uploading this very session back onto the schedule. So keep that, share it, and keep visiting it. So I think, Eugen, it's time for us to continue. Take it away.
Thanks, Indy. So guys, before we went into break, we looked at microscopes. what I've got a few images here, and you can see them on the page, and you'll see that this is the diagram of a red blood cell, guys. And we will be talking about red blood cells when we look at types of animal tissue later on during the course of the year. Here's the diagram of a nerve cell, guys. Very, very interesting. And here you can see the synapses between the cell body and the axons and the dendrites leaving. Okay, and that's it. And below that we have the diagram of a bacterial cell, and this is an E. coli, and these are rod-shaped bacillus cells, but quite interesting in their shapes, and these would need an electron microscope to be able to see that kind of resolution. And here we have an amoeba cell. This is a unicellular organism, and here you see it has some vesicles around it, and this is the shape of the organism, and it has the ability to move, and here it has engulfed its prey. So quite interesting to look at amoeba under a microscope. We're going to move on to types of cells. So I know in grade 11, guys, when you're looking at cells and you're looking at microorganisms, you need to differentiate types of cells, and I thought that it's important for us to identify two types of cells before we're getting into actually plant cells and animal cells. So when we look at cells, we can classify them into two groups: prokaryotic cells and eukaryotic cells. And simply prokaryotic cells would be cells that do not have no true nucleus. Okay, you will do much more detail in grade 11, but a eukaryotic cell has a true nucleus. It has a true nucleus. Okay, so you see a clear membrane-bound nucleus. Here we're looking at a prokaryotic cell, and this is an example of a bacteria, and here you see that it has nuclear material that is not membrane-bound, and it relies on these structures which are called flagella for movement, and here you've got ribosomes inside it. So it's quite an interesting diagram to look at. Again, I said that we're going to look at it in greater detail in grade 11, but it's important for you to understand the concept now. The characteristics of prokaryotic cells are that there is no true nucleus. They have strands of DNA or RNA. It's never both. They do not have structures surrounded by membrane. So they have structures that are very simple, very primitive, and that's why we call them pro, meaning from some of the first forms of life. They also have very simple internal structure, very few organelles, and they're very primitive in their development, and they're one-celled organisms. An example of that would be bacteria. Okay, we could look at the next type of cells when we're looking at classification of cells are eukaryotic cells. And when we look at eukaryotic cells, eu meaning true, and karyon refers to the nucleus. So cells that have a true nucleus. Here we're looking at a cell, and this is a plant cell, guys. And here you can see the nucleus. It has a complex cell wall. It has a cell membrane. It has, it's got mitochondria in here. It's got nice nucleus around there. And here you're seeing some vacuoles. So we see quite a complex development, and you see some other organelles in here. And here you see some reticulum, which has. So a cell that has lots of internal detail and complex development as compared to a simple cell. So the characteristics of a eukaryotic cell would be that it has a true membrane-bound nucleus. They have structures surrounded by membrane. So we looked at most of the structures, the mitochondria, the Golgi apparatus. These are structures that are membrane-bound, and the internal structure is quite complex. It's not a simple cell, it's very complex. And obviously something that is much more complex than a prokaryotic cell. So as I said at the start of the lesson, today we will look at animal cells. Next week we will look at plant cells, and then we will do a comparison between the two. But let's look at an animal cell in detail. I know Indy said to me that during the break, she said that this was probably her favorite section because there's lots of colorful things you can see in a cell. Here we're looking at a diagram with lots of parts. I'm sure you remember some of these parts from school, Indy.
I do. Yeah.
So, and the most important part is when we refer to a cell, and especially an animal cell, is that we look at the nucleus. Okay, and obviously you remember from grade 8 and 9, guys, when you did natural science, you probably looked at the function of the nucleus. So when we look at an animal cell, we see the different parts, and this has got the nucleus. So we see the nucleus, the nucleolus, and we see lots of mitochondria in here. We've labeled the mitochondria. Here we've seen lots of mitochondria, and we see some rough endoplasmic reticulum. We're going to look at each of these organelles in detail. So what is an organelle? Basically, this is a functional unit of a living thing. We call this a cell. In order for this cell to function, it has smaller organelles, or functional units that actually make it possible for this life form for this cell to function. So we refer to the mitochondria as an organelle. It carries out a specific function, and together with the other organelles, they function together to ensure that this organism or this animal cell can function. Okay, so that's a diagram of a typical animal cell. Here we're seeing a plant cell, guys, and here we're going to look at the structure in greater detail next week, but this plant cell has a definite shape. It has cell wall, it has a cell membrane, and as you can see here, we refer to the cell wall being made up of a cytoskeleton. So we're not talking too much on about plant cells today. We're going to go on to the details of an animal cell and see what the parts of an animal cell are. So the typical animal cell consists of the following parts. It consists of a cell membrane, a nucleus, the cytoplasm, and the various organelles. So we're going to look at each one of these parts. And remember, when you're studying these parts, guys, look at the part, look at its structure, and identify the function of that part. And that's essentially how you would learn the cell parts and the cell organelles. You would identify the structure. You should be able to label a diagram, provide its function, and talk about its structure and what it is made up of. And essentially, that is what we're going to look at. Most importantly, when we start off teaching cells, especially plant cells and animal cells, we refer to the differences between them of having a cell membrane and an animal cell not having a cell wall. So a key difference between a plant cell and an animal cell is the presence of a cell membrane only in an animal cell. So here I've taken an electron micrograph, guys. An electron micrograph is an image that has been taken using an electron microscope. We would not be able to see this with a light microscope. So it's a microscopic structure. We're looking at the details of the cell membrane. Now you've got to remember that whenever we see a cell, we obviously tend to see a plain structure, but a cell, imagine a cell is a complex structure. It's a three-dimensional structure. It has a skin of cell membrane around it. And when we look at that cell membrane, guys, this is the outer protective layer of the cell. It's important for us to understand the concept between cell membrane, because the next topics are diffusion, osmosis, and movement of substances in and out of a cell. And this is what controls substances entering, leaving the cell, and offers protection. It also is able to respond to changes in the environment. So we've got to look at why the cell membrane is a very important part in a cell. So if we look at the cell membrane, it is the outer membrane of a cell that controls the movement of substances in and out of the cell. It is a double layer. So it's a bilayer, and we're going to look at it very quickly. We're going to look at the structure. And we refer to this model, guys, as the fluid mosaic model. And a fluid mosaic model, and or probably you heard the term mosaic before, and mosaic basically is it's sort of a collage of different structures put together that give you this nice finish or image. And here we look at the structure of the cell membrane, and we see different structures put together. The key structures to be able to identify are the phospholipids, and I'm going to change the color of my pen so that we enhance this. So these are the phospholipids. Okay, and we refer to the cell membrane as a phospholipid bilayer. I'm going to read through the notes very quickly. The fluid mosaic model describes the structure of the plasma membrane. Different kinds of cell membrane models have been proposed, but one of the most useful is the fluid mosaic model, which I'm explaining to you here today. In this model, we see the membrane is seen as a bilayer of phospholipids, in which the protein molecules are embedded. So if we look at this image, guys, here we're seeing the protein molecules, and these form the channels through which substances enter or leave the cell. So this is basically the point of entry or exit of substances moving into a cell. So they control substances, very similar to what a door does in your house. You're able to open and close, but this is very complex in terms of the way it functions. We also see that the membrane is made up of a phospholipid, and we call that a bilayer. Bilayer because it is made up of two layers. So here we're seeing, here on this side we're seeing, they sort of seem like lollipops. It's got a nice head there, guys. It's a phosphate head, and it has a lipid tail. The heads are hydrophobic and hydrophilic, and they point towards the outer surfaces. The tails are the lipid tails, and they are hydrophilic, and they point towards the inside. Here we're seeing the membrane proteins, guys, and as I explained to you, and these are the membrane proteins that control the channel of substances moving in through. Here we're seeing on the surface of a cell a glycoprotein, and this is what is the recognition surface, recognition proteins on different cells, and that is how the body is able to identify different cells by these glycoproteins. So we refer to this as the membrane or the plasma membrane, that is the skin, so-called the skin outside the cell. Here's a detailed diagram showing you the channel, the protein channel, that shows the movement of substances into and likewise out of there. So this protein channel will, along with the energy from ATP provided by the mitochondria, be able to open up, allow substances to move in and exit. And here we clearly see the phospholipid bilayer. We're seeing one layer there, another layer here, and each layer, as I said, is made up of a phosphate head, and that's the phosphate molecule, and that is the lipid molecule. Okay, so it's a phospholipid bilayer made up of two phospholipid molecules. So the channels or the pores, the channel is in the cell's plasma membrane, is made up of certain proteins whose function is to control the movement of food and water into the cell. These channels are made up of certain proteins that are specific in their function, and they control the movement into and out of the cell. Okay, let's move on to the next part. Another important, now we move into the internal parts of the cell, and often we start with the most important, and this is the nucleus. And you probably know what the nucleus is, and it's probably one of the most important structures in a cell, because this is the brain of the cell, so to speak. It contains all the genetic information. It also controls hereditary information. So in terms of how does the cell function, it has a control center, and that control center is the nucleus. It will coordinate all the functioning of the cells and of the entire body. So in it we have the nuclear material, which is called chromatin network. And that chromatin network, guys, you often see a network of fibers that are entangled in there, and that's actually the DNA. The DNA is what makes us unique, and it's a code that is what the cell has that controls the functioning of the cell. So the nucleus is a control center of the cell. It coordinates the functioning of the cell, and it is the largest organelle in the cell. And it contains DNA, we said that. The DNA of all cells is made up of chromosomes, and chromosomes basically are the arrangement of the chromatin network into specific structures. We will look at the functions of chromosomes when we look at cell division later. The DNA is obviously what codes for life in a cell, and basically it plays an important role in cell division. The nucleus, you'll find that, is responsible for making also ribosomes. So it is responsible for sending out the code to produce proteins and also to make ribosomes. The circles on the surface, guys, these are pores. So we refer to these as nucleopores, and it is through these pores through which a pore is a microscopic opening, a gap that allows certain substances to leave the nucleus. The internal contents there is called the nuclear plasm, and that's where the nucleus and the contents of are bathed in. Outside the nucleus, we have the cytoplasm. We also have a nucleolus. We're going to look at the function of the nucleolus, guys, when we get back from break, and we're going to see how the nucleus and the nucleolus function in controlling the functions of the cell. Indy, over to you, and we go for a break.
Okay, guys, we're going to go for a little break. There are a few questions on the page. I will be going over them with you again during the break. So if you do have questions, please post them onto facebook.com/learnextra or on Twitter @learnextra. We are here to help you. And guys, there really is no such thing as a silly question, only the one that is asked after the exam, because then it's far too late. See you now after the break. Hi guys, and welcome back to Learn Extra Live. It is a Wednesday. That means that it is Life Science and Grades Day, and yes, you are still on. So we've got quite a few questions on the page, and Eugen has said that he will kindly go over them. So guys, you better keep them coming. You better keep them coming, because sometimes that's how you learn, is by posting these questions on the page. Also, if you see questions on the page that you know the answer to, please answer them, because you learn while you're teaching others. It's pretty awesome. Okay, shall we start with number one?
Okay.
This one is from Tam Sangkwa. I hope I'm saying your name right, Mindset. I apologize if I'm not. And he wants to know, Does bacteria photosynthesize?
Quite an interesting question, guys. I know this is a grade 11 question, but yes, there are certain bacteria that can, we call them chemosynthetic bacteria. We get them, that bacteria that are able to use chemicals to produce food. We have photosynthetic bacteria, and some of these bacteria actually have chloroplast in them, so they can photosynthesize as well. So you have your chemosynthetic bacteria that use chemicals to produce food. You have your photosynthetic bacteria that actually use light energy to convert to produce glucose. So, and you have your thermophilic bacteria that can live in extreme conditions of salinity and temperature as well. So yes, bacteria can photosynthesize.
I didn't even know that.
Okay.
Now I do. Okay, now this one's from Mameli, and he asked, true or false, is mitochondria passed down from your mother?
That's an interesting question. Again, that's a grade 12 question, and we get this in terms of when we talk of mitochondrial DNA. So when we looked at DNA, there are different types of DNA in a cell. We have, in the nucleus, we have nuclear DNA, guys, but also some of the organelles, like the chloroplast in a plant cell, has some DNA, as well as the mitochondria in animal cells has DNA. And the mitochondria has DNA is because the mitochondria needs the DNA, so the DNA codes for essential proteins and enzymes that are needed for cellular respiration. So the mitochondria are present in the DNA contains mitochondria. The significance of that is, I'm going to very quickly go down onto explaining to you the concept of how the question was, is only mitochondrial DNA passed on to the offspring? And that's true, and it's only from the mother. And this takes us back to a bit of reproduction. I'm going to very quickly show you a cell and assume that is an egg cell, and that has its nuclear material here, and that's a sperm cell, guys, and that's the acrosome, and it has its nucleus and it has the genetic material. The mid piece of the sperm contains the, I'm going to draw this in a different color, the mitochondria. Okay, remember that the egg cell also contains mitochondria. Okay, when these two cells fuse or fertilization takes place, they come together, you'll find that the sperm, the head of the sperm enters, and the head of the sperm contributes its nucleus, and the egg contributes its nucleus. The mitochondria comes from the mother, so we have the mitochondrial DNA coming in from the mother, and that's mitochondrial DNA. But the sperm, I haven't drawn the tail, so it's going to look a very funny sperm. The mitochondria breaks off with the mid piece, and it's only the head and the egg, the head of the sperm and the egg that fuse together, and these will become the nucleus, and that contains the chromatin network, and the mitochondria only coming from the mother. So essentially what this means is that your mitochondrial DNA is inherited only from the mother. She passes it on to the offspring, and the offspring will then, only the daughter will pass it on to her offspring. Okay, so when a guy marries, it's going to be his wife's mitochondrial DNA that is passed on to their kids, but his mitochondrial DNA can be used to determine his maternal ancestry. So that means he can work out who his mother is, his grandmother is, his great-grandmother, and so on. And that is called maternal DNA ancestry. It's done at the Origin Center. Professor Himla Sudial heads this, and she does mitochondrial DNA. The men can test mitochondrial DNA. Yes, you can, but you don't pass it down to the next generation, so that's a dead end for us. But if we can work out our paternal DNA, that is your Y chromosome DNA, which we inherit from our dad, so we can work out your dad, your grandfather, your great-grandfather, and so on. And there are marker regions, and that's quite interesting. I know Mr. Khan in grade 12, he does the mitochondrial DNA, and he'll be able to explain to the grade 12s in much detail in terms of, so that's quite interesting. And that actually helps us to determine our Y chromosome inheritance and your mitochondrial DNA inheritance.
That's incredible.
So cool.
That's so interesting. Okay, two more questions.
Cool, we'll take
two more
questions.
questions.
We must take these questions.
Okay, so now this one is from Theophilus, and he asks, does a cell have a specific size?
Do cells have specific sizes? Yes. What would you think the biggest cell is in the body?
Oh my gosh, I mean,
We just mentioned
tiny.
it now.
Oh, so the biggest cell.
The biggest cell.
Your
possible.
skin. Your
That's
skin.
the biggest organ.
That's the biggest organ. Ah, the biggest cell. Ah, now you're gonna have to tell me, guys. I bet you the grade 10s at home are probably shouting it to me.
Okay.
Indy, it's
the egg cell.
the egg cell.
Okay, that's probably the biggest cell in the body. Okay, but cells may vary in sizes. So we've got the red blood cell, which is so the red blood cells that I'll show you very quickly, guys. The red blood cell, this measures 7 micrometers in diameter. So that means if we measure from there across to here, I'm just going to change my pen so that we can see this. If we measure the diameter of this measures 7 micrometers in length. If we compare that to an egg cell obviously is much larger than that, and that could measure up to 150 micrometers, and it can increase in size. So yes, cells vary in size, and that's because of their function. So the egg cell obviously has this is an anucleated cell, doesn't have a nucleus, and it's much smaller. These are nucleated cells, they have a nucleus, but it's haploid, so it has half the genetic information. So our cells will vary in sizes. And it's basically looking at our skin cells. These are flattened cells. They are much narrower than broader. You have your cuboidal cells, your cube-shaped cells. So throughout the body, guys, we have cells that are specialized to perform functions. And according to their specific functions, their shape and their dimensions are accordingly adapted. So all our cells do not measure the same. They can be extremely small to being extremely large. If we look at a nerve cell, which is much smaller than a red blood cell, so that gets much smaller. So definitely cells are extremely small to being much larger, but we will still need a microscope to see an egg cell.
Okay, so it's not like we have huge cells.
No.
Yeah. Okay, so now I think this one, it came from 2 different learners, but let's ask anyway, and it's the last one, and then we're going to carry on with the show.
Okay,
cool. So now this one is from Vusani, and he asks, we only have 2 types on a microscope, and what is the function of fine focus screw?
Okay, so I'm going to get back to that slide very, very quickly, guys. Okay, so we have 2 types of adjustment on a microscope. Basically, it's called a fine adjustment and a coarse adjustment. And so here, when we look at this diagram, here we're seeing a coarse adjustment screw, and that's basically to move the stage. So when we use the coarse adjustment screw, we move the stage and we bring the specimen into view. Now it's about bringing the specimen into view. Once you establish that the slide is, you can see some amount of, you can see some organism, or you can see your specimen, you would then now use your fine adjustment screw to be able to differentiate between the different views. And obviously, and that is based on, think about looking at using a camera, guys, and when you need to focus pictures in focus and out of focus. So that's essentially what the fine focusing does. And often when you're using even your phones, you've got that button where you press and you hold, and then it gives you the green flicker to say, yes, you can click. And that's basically the fine focusing. So, but this is done manually, where you now turn the fine adjustment very, very carefully so that you clear the acuity of what you're looking at. And that actually helps you to differentiate different parts. And remember that the parts that you're seeing have a dimension, and at a sub-microscopic level, guys, the difference between looking at this and a layer below it would need fine adjustment to be able to differentiate what's on the surface and what's below. And that's the reason why you have a fine and a coarse adjustment. So
Fantastic.
Cool. We will take questions. I like the questions coming, and that's what we should encourage, guys. But it should not distract us from finishing what we're doing. So we're going to get back to content. And I'm going to jump very quickly to this interactive site that I've been talking about. And I know that there were questions about the Golgi body, but we're going to go, guys. So if you log on and you looked at this, so we looked at the nucleus, guys, and this is quite interesting because you can actually play around with this. And you click on the nucleus of a cell. I'm going to go back to the animal cell and show you this here. I'll show you the animal cell very quickly. Okay. Animal cell. Okay. Let's look at the entire cell. Okay, so that's the nucleus. When we look at the nucleus, guys, here we're seeing the nucleus. Okay, in it we spoke about the chromatin network, and here's an electron micrograph showing the internal structure of the nucleus, and here you can see some chromosomes, and this is a cell, if you guys can see, this is a cell that is undergoing cell division. Okay, so that was the nucleus. We're going to very quickly jump back to my presentation, and we're going to look at the next structure which we were looking at. Okay, so we've gone through that slide. Okay, I'm going to very quickly find the slide for you. Go through there. Okay, we were looking at, the mitochondria. So let's look at the mitochondria. And the mitochondria, guys, is basically, it's an oval structure. The function of the mitochondria is, we often teachers use the word, it's the powerhouse of the cell. It's a double membrane structure. So here you can see this is an electron micrograph, and it's got these folds, guys, that create a very sort of a stepped feature. Some of us compare it to a walnut. Okay, but I'm going to show you an illustration of it. And here you see these folds, and often easier to be able to explain from this and then refer to that. So here we're seeing an outer membrane. It's a double unit membrane, so we can see it has 2 layers. And so it has 2 layers right here, guys. There's 2 layers. It's a double unit membrane. And it has these folds, guys. And these folds are called cristae. And these cristae are there to actually increase the surface area. It's about compacting more information, compacting more membrane inside so that enzyme activity can take place. And remember that the mitochondria functions in producing energy for the cell. And in it a little earlier on we had a question about mitochondrial DNA. So we have, in it we'll have circular structures which are mitochondria, which are mitochondrial DNA. And we also find we have ribosomes present in it to produce protein. So I'm going to go back to the mitochondria electron micrograph to show you what it would look like under an electron microscope. So it's an enclosed structure, and it's distributed throughout the cytoplasm. And the main function is that it's responsible for a process called cellular respiration, in which it produces ATP. And ATP is the energy molecule in a cell, and you need glucose for that. Every type of cell has a different amount of mitochondria, and that is quite interesting. What's important to remember is that mitochondria will be in larger concentrations in cells that need to produce lots of energy. So if you think about in your body, you've got muscles that are the center that produces energy for your body. So if we looked at a muscle cell under the microscope, we would see that these cells would have a higher concentration of mitochondria. If you looked at the wing muscle of a fly, you'll find a dense concentration of mitochondria. If we compare a skin cell, we'll have very little mitochondria. And that's because we don't need much energy in the skin. So depending on the concentration of mitochondria in the cell, you'll find that distribution would be affected, would influence where the location would. Our heart muscles, guys, the heart's pumping all the time. So you find that your cardiac muscles have a high concentration of mitochondria, and that is to produce lots of energy needed for respiration. Okay, so we looked at the structure of the mitochondria. We looked at the inner matrix. So it has an inner matrix which is fluid-filled, and it has this elaborate, it's sort of more like a labyrinth of folded membrane called the cristae, and in it we have enzyme activity taking place, and that is the enzymes that are required for the process of respiration. Okay, we're going to very quickly look at ribosomes. We're going to introduce the concept of ribosomes, and then we're going to look at the structure and details after break. But ribosomes, guys, these are the organelles that are basically responsible for protein synthesis. So these are the protein factories, as to say. So this is where your proteins are made. So if you think about the amino acids that are needed in the body, the amino acids are put together by your ribosomes, and that is called protein synthesis, and that takes place by the organelle called ribosome. So we're going to go for a break.
It's the end of the show. Can you believe it?
Is it?
Yeah, Eugen reckons he's got another hour. Should we carry on? No, the director says no.
No. Okay.
1 minute left.
It's amazing.
I'll take 20 seconds. You can take 30.
Okay. So I'm going to quickly jump to the questions, guys. And I think it's important in any topic that we look at questions when we're studying. We often get carried away, and I think it's important for us to revise at the end. I'm going to quickly jump to questions. Remember that these notes are there for you. I've put in some interesting questions here, and I want you guys to answer these questions. And maybe you can post some questions. You can post the answers on the page, or you can interact with us next week.
You know what I'll do for you, Eugen? Guys, okay, we've come to the end of the show. But it's okay, because what I'm going to do is I'm actually going to post these questions on the page. I will make sure that I take screen grabs, and I take screen grabs of all of those questions, and we'll go over them next week. What do you reckon?
Cool. That's a good idea.
Fantastic.
Yep.
I think that's a fantastic idea. Guys, these notes are up on our website. What you need to do is you need to go to www.learnextra.co.za /live. Go and find today, click notes, download PDF, and that is just as much, well, that's how easy it is. Thank you so much, grade 10s, for making the show amazing. You guys have been incredible. Grade 11s, you are up next. You guys better stay tuned for the next show. Meet one more of our top 10 presenters, a teak. Thank you guys, grade 10s. You rock. See you next week, same time, same place. Bye.